Hey everybody, my name is Taylor Fulk, and today we're going to talk about exercise 1, how to observe and describe biological specimen. And this is going to be covered in the Bio 1802 lab. So without further ado, let's go ahead and get started. So these student learning outcomes are a great way to check in with yourself, to know where you're at with each exercise. So they're going to talk to you about what you will know and understand and what you'll be able to know and understand, as well as what you'll be able to do once we finish going through the exercise at hand. So if you find yourself a little perplexed by any of these topics or concepts, please reach out to your instructor and let them know, and we would be happy to help you further your understanding on that. But these are a great way to check through the main points that we're going to hit and make sure that you're ready to move on to exercise 2. So we'll start by just covering biodiversity. So biodiversity is a term that's used to describe the number of species in a given ecological area. So biodiversity, when there's a lot of biodiversity, it actually increases the hardiness and resilience of the surrounding ecosystem. So when there's more species, it will actually give a stronger ecosystem. When there's less species, you're going to have a weaker ecosystem. But why is this? So how this works is when there's multiple species, let's use plants for instance. There's multiple species of plants in an ecosystem. We can also talk about multiple species of animals, fungi. All of these things are contributing to that ecosystem. They all have a role to play, and that will increase their productivity with more diverse abundance of those. So it's really important to note that biodiversity is not only dependent upon the numbers that we already have, but conserving the biodiversity that we have here on Earth. So this seems like a good transition to talk about extinction. So you hear a lot about, all these species going extinct in the news, and it is alarming, especially because the background rate for extinction is about 1 to 5 species per year. So extinction is a natural phenomenon at that rate. However, we're surpassed that at this point. We are now losing species at 1,000 to 10,000 times that background rate. So this means that literally dozens of species are going extinct every single day. So here we have a conceptual diagram to help us visualize how this works out. So each rectangle here represents a plant community containing individuals of either blue or green species. So here we can see that this is going to be community 1 in the blue and then community 2 in the green. So on our first year here, we're going to document that there's average temperatures going on. So it doesn't really look like that's affecting either community so far. However, when we move over to year 2, it seems that the green species is being favored over the blue. So the community that contains these green species here is actually thriving in these warm temperatures, while the blue, their numbers are kind of dwindling. If we transition to year 3, the cooler temperatures look like they're favoring the blue species, while it's having the opposite effect on the green species. So this is a very natural phenomenon of interannual climate change that happens where the temperatures go up and down. And when communities don't have high biodiversity, say with more than one species, it's going to be hard for them to be productive. So to tie it all together, a community that only contains a blue or a green species is going to fluctuate in that biomass where there is interannual climate variability. However, in contrast, in a community containing both green and blue individuals, the decrease in one species is going to be compensated for by an increase in the other species. So this will actually help stabilize the ecosystem productivity between years by having more than just one species that's responsible. So it's also important to know that on average, the diverse community will exhibit higher productivity than either of the single species communities. And that's pretty self-explanatory. So if you have um, these individual communities here that you see on the bottom two, and their numbers might dwindle depending on what type of climate is offered that year, then they're going to have a hard time being productive. But if you have another species in the loop, it's going to be able to compensate for the lack thereof. So we do have a vast amount of biodiversity here on Earth, and with close to 8.7 million eukaryotic species, which doesn't even include the prokaryotic ones, which we will touch on, and they have an amazing amount of variation. We have a lot of work to do if we want to conserve all of these species and keep up the resilience of our ecosystem. So first things first, determining whether or not a species is being discovered, if it's a new species or maybe visually it looks like a different species, but it's just a different variation. This happens a lot with owls, and they molt. So, you know, when that was first happening, I'm sure that was really confusing for the scientists at that time. And also freshwater mussels that are endangered, and a lot of them look really similar, so it may be even difficult to tell them apart. And then we go into naming these species. So coming up with scientific names that are distinct to that individual organism that no other species has. And usually these names are going to be formed upon by their ecological habits that are studied by scientists, as well as what role do they play in the ecosystem? What benefits do they give back to the ecosystem? And yeah, so all of these things, as well as possibly putting together a conservation plan. So what the conservation plan is going to do is ensure that these species are not being disturbed in an unnatural way. And by conserving them, we're not only, I would quote, saving them, we're saving the ecosystem in itself, because it's all a system and they all work together within the ecosystem, and they play all these roles that are important to the function. So the implementation of these conservation plans is really key. This comes in at a government level, and it can get sticky because then we start dealing with politics. But the research that goes behind these conservation plans are really the future, and they're really what is going to save the biodiversity that we still have here on Earth. So when we get into the naming of these organisms, it's going to be based off a field called taxonomy. So taxonomy is an internationally shared classification system. And this is represented here on the right with this upside down pyramid we have over here. So we have our domains at the top, and it's where it's going to start as the most exclusive. So it's going to contain all of these different species. I'm sorry, most inclusive. Totally fumbled on my words. So domain is going to be the most inclusive. It's going to contain all of these different species and the largest amount of different species and organisms that will fit into it. So if we panel down to the kingdom, it's going to be less inclusive as we work down the line. So it's going to get more and more particular. And the further down we get, it's going to work its way down to being very specific and get down to the species level itself. So the way that we would name the species is by using the method that we call the binomial system. And we'll talk about that and why it's important and what it used to be, because there used to be another method that wasn't so nice and wasn't so clean and to the point. But for the purpose of this, you can see that we have domain, kingdom, phylum, class, order, family, genus, and species. And this is going to come up a lot in Bio 1802. So it's really good to hit the ground running with a great understanding of how taxonomy works to set the stage for a great semester. All right. So if we look at this in more detail, I personally love this diagram. I think the previous one does a very good general explanation, but this is so visual and it really allows me to apply what I talked about in the last slide in picture form. So let's get started on this. So we'll start with the domain, and you can see that this domain, and we're using eukarya here, and this encompasses eukaryotes, and we will definitely touch on this semester. But we have a variety of different species of plants. We have mushrooms, cacti, angiosperms. Looks like we got some lizards, some reptiles. We've got a vast variety here, very diverse and not really specific, right? So when we drop down to the next group, we're going to be classifying this as the kingdom Animalia. So animal, haha. And it does fit in the domain of eukarya. But now we're going to be a little bit more specific, okay? So you notice that on the left side, we've dropped all of the plants and we've dropped all of the fungi. So now we really just have animals. And then if we panel down one more to the phylum, we see that we're investigating the phylum Chordata. So the phylum Chordata, by definition, it includes animals with a notochord, a dorsal nerve cord, pharyngeal slits, and a postanal tail. So as we work our way down here, going from phylum to class to order to family to genus to species, you can see on this right-hand side here in the black words, sorry, in the black words on the right here, it's telling you what each step down is getting more specific with and what this certain classification has to have in order to be as specific as it gets. And when we get down to the bottom, as specific as it gets, we have the gray wolf, which scientific name is Canis lupus. So that's pretty much how it works. And we're definitely going to be touching on this more over the course of our semester. So prepare yourself now and become very familiar with it. So why do we use binomial nomenclature? So that's going to be talking about the genus and the species. And to name these species, I'd love to talk a little bit about that history. So before 1753, that was a long time ago, plants were named using diagnostic phrases that often became these long phrases, and they were continuously subjected to change. So if we look at the old nomenclature here for this beautiful organism in the background, and it's called the elderberry, common name elderberries. If we look at this old nomenclature, which was classified in 1740 as seen here, you can see that it almost looks like a sentence. And it's a very old method that was time consuming. It was exhaustive to try to remember the length of that name. This name in itself means branch trees, flowers, and stems arising. I mean, that's basically a sentence. So when we think about names, after 1753, thankfully, we switched over to a much more effective method, which is called binomial nomenclature. So while we used to have this multi-word, what they call name, we now have something much more simple, to the point, but it doesn't miss out, on the details of the organism itself. And it will be in Latin, which is very convenient for us because Latin's a dead language. So Latin is not subject to change, as is modern language. So the name in itself will never change. So once you have a scientific name memorized, you should be good to go. So although we don't really speak Latin, it is very beneficial for naming in a scientific field. Another reason why we use binomial nomenclature is the clarification. So each plant has a taxon specific name, often hinting towards these morphological traits. So you can see the color of this elderberry is a very dark color. So the specific epithet here, the second word for the binomial nomenclature, which we'll get into in just a moment, is nigra, and that means black. So this is an example of how that helps clarification wise. So it's also placed into categories that share those same traits, as well as giving us insight to relationships that infer names based on recognition of their physical characteristics. And again, touching on the fact that this is a universal way to classify organisms. So I think of math as kind of like, it doesn't change across languages, right? It's math. But we can also think of scientific names and binomial nomenclature as something that's universal, and it won't change because we're using Latin. So binomial nomenclature does follow three things. So three main points we want to touch on here. The species name of an organism consists of two parts. So that's going to be the genus and the specific epithet. I mentioned the specific epithet in the last slide, and we're going to touch on that now. So the genus is going to be capitalized, while the specific epithet is always going to be lowercase. Not caps lock on the genus, but just that first letter of the genus is going to be capitalized. So both the genus and the specific epithet are presented in italics. And if you're handwriting these, they're going to be underlined. So notice I said specific epithet and not species. Okay, so here's an important thing I want you to know, that genus and specific epithet combined are a species. That name that you're going to write down is the species. So I thought we could kind of play with it here. We have this really adorable bird here, and we're going to practice how one might go about naming it. So it looks like this bird has a pretty small mouth, right? So another word for small is going to be micro, and mouth is also called stoma. So something we might start off with would be microstoma. We can also notice that it has very distinct tail feathers. Specifically, it has 10 tail feathers. So decim means 10, cauda meaning tail, and pluma meaning feathers, like plumage. So decim cauda pluma. So microstoma, decim cauda pluma, gives you the possible binomial name. And you can see here the correct way it is typed out. If this were written, it would be underlined. Just want to note that. But we can see the capital letter for the genus and how the specific epithet in itself is going to be lowercase. But together, that makes the possible species, excuse me. All right, so if we break this down, the genus that we just went over is going to be the microstoma. Again, I can't stress this enough. We're going to capitalize the M here. Always capitalize the first letter of the genus. The specific epithet, decim cauda pluma, it's totally okay to say it in syllables, all right? These are really long words. And honestly, I couldn't get by if it wasn't for Dr. Osborne, who, while in herpetology, I was trying to learn all of these taxonomic names, and he was like, just break it up. And I was like, well, duh. But it's not something that I really thought about. I was sitting here trying to say it all at once. So save yourself. Um, but together, when we put microstoma, Decimcata pluma together, we have the species. And this is how you would see it written out. Typed out. Written out, it would be underlined. So let's talk about drawing specimen and how that's done properly. So it's going to be a very important skill that we use throughout this semester, but it's also just good to know for any type of learning that you're going to be doing in the future. So biology in itself is going to be full of information, lots of specimen, complex patterns, extraneous cell structures, and details. So while all of this feels important to document, it may not be important to document it all for the task at hand. So our task at hand here is going to be identification. And one of the most important things about being able to do an effective and quick drawing of a specimen is to distinguish, well, what is important? What is the most important information that I want to put forth to this drawing? So why even draw something when my phone can do that? That's a very good question. Considering the mobile world that we have these days, it seems much more convenient. But when we draw something, it forces us to require time and attention to detail. We learn things better when we draw them. It's just scientifically proven. You have to think about the scale of something. So for instance, we draw in this lab a male pine cone and a female pine cone. A male pine cone is about this small, and a female pine cone is what you're going to be the most familiar with, and they are about this big. So when you draw it out, it's going to make a huge difference to recognize the scale of that. Wow, if you're up close with your phone taking a picture, it could look like the same size. So, drawing is a type of active learning that has been shown to foster observational skills, memorization, understanding of spatial relationships, so construction of a mental model, um, connections of their concepts and their ideas. It helps you process data better, and it gives you a better understanding of the big picture or the core concept. And lastly, I want to say that it can distill the amount of information captured. So this is going to be very important for your worksheets this semester. So an effective biological drawing is going to be composed of 4 parts. The illustration itself, so we can see here the drawing. Let me click my little pointer here. So showing you the drawing itself. Not too much detail, but it's still enough detail where it needs to be. And you'll get better at this as we continue to draw this semester. So, and your instructor should give you good feedback on how to improve your drawings. So the labels themselves, we'll talk about that now, are very important. But we are often going to cue you with what labels you need to put. So there won't be really any guessing game as to what's important to label and what's not. And I think this arrow is a little off, but we're referencing the title. It's pointing at the title, but it's referencing number 4 here, but we're talking about the title. So we always want to know what we're looking at, just in case your drawing isn't going to be exactly like what you looked at, right? Or you want to pan back to your drawing. It's always good to include the scientific name and the common name in your drawings. And lastly, I want to talk about the notes. So the notes are often confused with labels, right? Or vice versa. But the notes are really there to distinguish this organism from another. So maybe you're looking at a protist under the microscope and you notice that it's motile, that it's moving. And of course you would label like a flagella on your picture, but it's going to make a lot of difference if you say was spinning in a corkscrew-like motion or an undulating motion. And you'll know why in our future exercises. So those little things that you can include in your notes can be of utmost importance when you're differentiating it from something that is really close or similar to that. It can also be great to put down like, hey, this was the color green, you know, because we won't be representing our drawings with color in this class. And most of the time in the field, you won't be doing that either. And one thing that's not on here, as far as just pointing at it, I want to touch on the magnification here. So whenever we're drawing stuff that is under a microscope or a stethoscope, a stereoscope, not a stethoscope, a stereoscope, we want to make sure that we're including that magnification. It's an important aspect, especially relative to size. So you will always be expected this semester to include these components of a biological drawing every single time. So if something is missing in your worksheet, like labels, for instance, we're going to have to take points off. And a title, you know, just as important. All of these things encompass what makes a biological drawing. So here's a great example. And we have E. coli pictured here. And you can see how we didn't include the shading, right? Shading is not something that we want you to worry about. It's very time consuming, and we don't want you to sit hours trying to get the shading just right. That's what the notes are for you to say, this is a little dark, right? So we have a really efficient drawing with the main structures, but also gives us the filaments, so we can definitely see those filaments extending out. We've got the magnification accounted for, and again, the title, labels, notes, looks great. So this is a great representation of what we want to see on your worksheet. Another great representation here. So if we were looking under the microscope and we saw this copepod, it's a type of plankton. This might be what you would draw in your notebook. So it's got all of the correct labels here. Doesn't have any of the shading, right? But the correct name is listed, and of course it's italicized because I typed that out. But if you're writing it, it would be underlined, but it's got the genus and the specific epithet. I almost said it. The genus and specific epithet to give us the species with the genus capitalized. So I also included the scale, which is going to be an important part of your drawings as well. And I want to go over why it's important. So we can have two very morphologically similar, there we go, bunch of words, bunch of syllables. So two very similar looking buttercups, but the distinction between these drawings is going to be key here. So they look very similar, but you can notice that when we get down to the drawing aspects, they're actually different species. So the way that these drawings differentiate between what labels might separate these species of buttercups is key. The notes are also important. I don't see any notes on this one, so just note that that is also going to be important as well. So one thing that is included here is 1 of 5 petals. So you might not have to draw all of the petals, but it might be a quicker way to just say, you know, what they did here, 1 of 5 petals. It might be also to distinguish between the sepals here. You see these sepals are representing a fold, while these sepals here are simply supporting the petals. So maybe that's something that you mention in your notes. But anyway, I think this is a great drawing to reference back to in case you're having trouble during your first drawings on your worksheets. So now on to dichotomous keys. So dichotomous keys are going to be extremely helpful tools that we use in biology, not just in this class, but in your future biology courses, especially if you're going to go the eco route. So they're really helpful for field biology and using them to identify organisms in the field, outdoors. They're also referenced as like field guides or identification guides, whatever you want to call them. So this is going to be based solely on physical characteristics. This is not going to be anything with their genetics or molecular characteristics, not any type of fossil records, like phylogenetic trees or anything like that. And dichotomous keys are not here to indicate any kind of evolutionary relationship. But the best way to start out a dichotomous key is with a broad, generalized characteristic, right? So the characteristics are going to get more and more specific as you move through the key, kind of like what we talked about at the beginning of exercise 1, right? So eventually finding characteristics that are specific to that individual organism. It's always going to be two-choice system. So always remember this or that. Do not try to include 3 or 4 or however many different scenarios here. It's going to be a this or that system all the way down. Yeah, and another way you can look at that is, does it fit into this category or does it fit into that category? And that's shown here on the right. You don't see any representation of any more than 2 this or that questions. And sometimes it does end and you're already done classifying, as you can see on the top left corner there.